Hello, everyone. Good evening. Welcome. I hope you're all doing well. My name's John Parker, Chief Exec at the Arboricultural Association, and I'm delighted to be able to welcome you to our Wednesday webinar for this evening. Hurrah! Please use the chat to say hello. If you have any questions, do pop them in using the Q&A panel. Please don't put too many questions in the chat because I'll probably miss them. This evening, you'll be hearing Ted Green and Lynne Boddy and friends talking a load of rot. Others will be joining the fun as we go along with a panel of experts out there ready and waiting to give you their thoughts and to hear your thoughts, because we want to hear from all of you too. So please do contribute via the chat. And if you feel the urge to join the panel on screen, then you can try and do so by raising your hand using the raise hand function. Don't just sit at home in your room doing this because we won't be able to see you. The raise hand button. And we will try and work out how to promote you as a panelist so that you can have a chat with our panel. The format is going to be pretty much the same as normal. Lynne is going to give a presentation to kick things off, and then we're going to get stuck into the discussion. The plan is to draw it all to a close at about something resembling 7:30, so about an hour and a half from now, and we'll see how well we do for timekeeping. If you enjoy this week, then you're going to absolutely bloomin' love next week because it's more of the same. We've decided that there's just too much to discuss here. It wouldn't be sensible to squeeze it all into one webinar. So please consider this is part one, and part two will be at the same time next week with some of the same faces, I'm sure. The link to register for that one will be in the chat as we go along. One additional piece of very exciting news. At the end of this week, all those of you who are association members will be receiving a copy of this document through the post, along with your ARB magazine, the brand new Arboricultural Association Strategic Plan 2022 to 2024. This was produced by our staff team and trustees. It sets out our vision for the next few years. We really like it a lot. We're very pleased with it. We hope you like it too. There'll be copies being posted out to everybody, and then also it'll be available on our website. So please let us know what you think. And there's a big section in the back all about how to get involved. So please be inspired and get involved because we need you too. Right then, on with the action. I'm going to take a bit of a gamble now, and I'm going to hand you over to Ted, who is then going to introduce Lynne Boddy. So sit back and enjoy yourselves. Ted, it's over to you.
Yeah, well, hi, folks, and thanks to the AA again for allowing some of us laymen amongst you. And really, it's nothing to do with me. They put my name up there, but in actual fact, this is Lynne's night, and it's something I've waited for many years. In fact, two of the participants in the original Talking Rock you probably can see on your screen, which is Caroline and Mike. And they set up a meeting for Alan Rayner in 1993. So let's face it, some of us laymen have a lot of patience before we could get to Lynne. And so I don't really want to say too much. I'm trying to cut it down. But really, the idea for me of getting Lynne off the ground and all the rest of it was to talk about how we actually started some of these things. We've got the Caroline and the Mike Ellison Day, which was incredible. And then we started the Ancient Tree Forum, which was primarily tree people, but a lot of ARBs. And a lot of those ARBs are still, if you like, in the focus of what we're doing today about old trees. They really are. They're still, and there's two of them, three of them in the audience right in front of me. So, and I go back to what's really touched me. That was the Ancient Tree Forum for several years, where we went out, had events all over the country, looking at trees. I know the regional AA did it as well, but we also got to other members of the public. But we've now gone back seriously to just arboriculture, and that is through the Thinking ARBs. And that really kicked me off was because the first Thinking ARB Day was at the Ankerwick Yew, possibly the most important tree for civilization, actually. And I'm not to go on to it about it, but what impressed me was when I asked people if they would come and look at the Ankerwick so that we could gain some experience, offer some advice, push things along. arborists on a Saturday turned up from all over the UK. And that really got me. I thought, these people are giving up a Saturday to come and talk to other arbs about a tree. And that has been some of my rocket fuel for all my life, thinking arbs. So then Reg and me moved on and started the Thinking Arb Days, where we were trying to have a theme. And of course, the theme goes out the window, the first tree you look at. But we do intend to have a Thinking Arb Day in Blickling in Norfolk in mid-March. And basically, it's to look at layering. But it doesn't mean to say that throughout the winter, I can twist John to have some of our Thinking Arb themes a bit when Lynn's finished. And I've already said to Lynne that she's got 4 evenings if she wants to, because to me, what Lynne says is basically fundamental. So Lynne, go for it. Please go for it. Otherwise I'll go on for hours. And I can't read my own writing anyway.
Well, thank you very
much. That's Lynne. There she is. Many of you know her. She's got a wonderful book out. And if you haven't got it, you should have it. And there'll be questions at the end of the next session to see whether you bought it or not. Lynne.
Okay, so as John already said, this is part one of a two-part discovery of fungi and trees. In this first one, I'm not really talking so much about trees, but giving you, as Ted said, basic information on fungi, things we need to know about fungi, and to give us clues as to how they get into trees and how they interact with trees. But let's see what happens as we go along. So I'd like to start by reminding you that every year land plants make vast amounts of new organic matter by fixing carbon dioxide from the air using sunlight energy. And this is what drives our terrestrial ecosystems. Everybody knows that plants are important, but of course, every single one of those plants really is dependent in some way on fungi. 90% of plants, or roughly 90%, have mycorrhizal associations with their roots, which feeds them with water and nutrients. And all plants are dependent on fungi for decomposing their dead organic tissues. And the amount of organic matter that decomposes is similar to the amount that is produced by photosynthesis every year. So huge amounts are broken down by decomposers. So let's have a think about what fungi feed on. And I suppose that they feed in 3 broad ways, 3 broad categories. There are the rotters, which are called saprotrophs, and they use dead stuff. There's some examples on the left there, for example. There are the killers, and they're called necrotrophs. And we can have as our example here, we have Armillaria species, and this is ash dieback caused by Hymenoscyphus fraxineus. There are also those that feed on living tissues. These are called biotrophs, the bio bit effectively meaning the living part. And I guess instantly you'll think when we say feeding on living tissues, oh, they must be harmful. And indeed, some of them are. Some of them are parasites, so things like the rusts and the smuts. They're much more subtle in a way than the killers who just go in and kill. They have a much more balanced relationship. And indeed, sometimes they keep tissues alive for longer than they would be otherwise. At this time of year, you often see green islands on leaves. These green islands are where the fungus inside the leaf is keeping the leaf tissue longer when the rest of the tissue is taking on their autumnal colours and dying, and they keep these tissues living so the fungus will be fed. But they are parasites, and they certainly affect plant growth, fruiting, etc., etc. But of course, where the real importance lies is with those beneficial biotrophs, those fungi that have a mutual relationship with photosynthetic organisms. Now, lichens are fungi which link up with green algae or cyanobacteria, but the one that we're really mostly thinking about is the mycorrhizal association, of course. So mycorrhizal fungi are biotrophs. They feed in conjunction with living roots. They get their sugars from the tree or other plant, And of course, in return, they give water and mineral nutrients. But as humans, as biologists, we often like to make these categories, but of course, the fungi don't fit into those categories spot on. There's overlap. So, for example, necrotrophs that kill not only feed while they're killing, but they feed as decomposers after the death of whatever it is they've killed. And the two examples I've chosen here are Armillaria and Heterobasidion. Indeed, Heterobasidion really can kill to get into trees, but usually behaves more as a heart rotter and it kills living cells, but it doesn't necessarily kill the whole tree. So remember that killers don't necessarily kill the whole organism. They might just kill a few cells as part of their way into dead tissues or indeed out of those tissues. Biotrophs also have other feeding abilities. So, for example, ectomycorrhizal fungi, those fungi typically associated with the roots of trees, often have the ability to do some sort of decomposition at any rate. It's not going to be as intense as decay by the saprotrophs, and still the fungus, the biotrophic ectomycorrhizal fungus, is fed sugars by the tree. It gets its carbon source from the tree, but it can do some decomposition too, typically to get hold of nitrogen, phosphorus, and things like that. And a couple of examples are Paxillus involutus, which does something called Fenton chemistry. I'm not going to talk about that today, but it's the way in which brown rotters rot. So it's a way of breaking down substrates without using enzymes. And Paxillus involutus evolved from brown rot fungi, and so it's perhaps not surprising that it has some abilities to do some sorts of brown rot using Fenton chemistry. And indeed, Cortinarius lactarius russula evolved from white rotters, and they can obtain mineral nutrients in this sort of way as decomposers as well, to subsidize other mechanisms. I also wanted to mention endophytes because they're becoming increasingly important, or at least I should say we're realizing increasingly that they are important. Endophytes are fungi, or indeed any organism, I suppose, that is found within a plant, and it is symptomless. We don't know from the look of the plant that there's a fungus or bacterium or whatever there. So symptomless endophytes. Some of them are most certainly beneficial, and this is a table from the book indicating some trees, fungi, and the chemicals that those fungi produce and the effects they have on certain organisms. So, for example, they may produce chemicals that are inhibitory to various bacteria, to autopathogenic oomycetes, etc., etc. Some of these endophytes, some of these fungi within plants are pathogens in waiting. In other words, given the opportunity, they may start to be pathogenic, and certainly a lot are decomposers in waiting. And that's absolutely true of those fungi which are in the trunks of trees, in xylem. Many of them are decomposers in waiting. Some are just there incidentally. But anyway, we'll concentrate on that a little bit more next week. Let's think more about decomposers. It's not only fungi that are decomposers, I should remind you. And the decomposers are different sizes, from microscopic at this end of the scale to definitely macroscopic at this end of the spectrum. Their size is indicated along the bottom. So we have bacteria which do have decomposer ability, or at least some of them do. Of course, the fungi, which is our main concern today, illustrated here with Hyphiloma. Nematodes, there was one wriggling down there just a moment ago, attached by a trap from a nematode trapping fungus. Protozoa, these are all microscopic. Then there are the mesofauna, things like the mites and the springtails, that's Acaria and Colembula, and of course the bigger things too, like the mollusks and the beetles and woodlice, etc. But of course, fungi are the most important decomposers in most circumstances, because they produce hyphae, these fine filaments that can get into solid objects. They make a vast array of enzymes. And some are able to break down the most complex naturally produced molecules in nature. That's illustrated here by this diagram of lignin. Please don't think that all lignin is like this, because you can see that there are lots of different components to the lignin molecule, and they're bonded together in different ways. There are all sorts of different lignins. We should call them lignins, really, both within a tree and certainly between tree species. Now, saprotrophic fungi, if we took them as a whole, can break down all naturally produced organic compounds, I guess. And so that's from the complex lignin to things like dung, petioles of leaves, and indeed, in this case, horn. But fungal species vary in their ability to break down different compounds. Some have very narrow abilities, only being able to cope with simple compounds. Other ones much broader abilities, like the white rotting basidiomycetes. And just to remind you, of course, that fungi feed by external digestion. So we feed by eating food, goes into our gut and is digested in our gut. That's internal digestion. Fungi don't do that. Their hyphae secrete enzymes, although there are non-enzymic mechanisms as well, which I have touched on already. These enzymes break down big molecules into small molecules, and the small molecules can then be absorbed. So that's the basic way they feed. Of course, there's a danger in this sort of mechanism in that the fungus might break down big molecules of wood. The small molecules are lurking outside, which could then be used by any other microorganism that's in the vicinity. And undoubtedly that happens. Okay, just to remind you then about the different types of wood decay. We usually think about broad categories of white rot, brown rot, and soft rot, but let's think first about stain. We often see wood that is stained. It's not the colour that you would expect it to be. And often this is as a result of fungal activity. Here's just one example, in this case, green staining by Chlorosplenium aeruginorescens, that you can see fruiting here, the ascomycete. Of course, staining often is a dirty, darker colour and not much liked by people who are growing trees for timber or for paper. But anyway, the point about stain fungi is that they really can only use relatively simple compounds. They don't cope with the celluloses and hemicellulose and lignin. They live on the contents they feed on the contents of recently living cells. And so we would only see them really relatively early, I guess, in the decay process, or perhaps at very, very late stages, maybe. Then there are the brown rot fungi, of course. I've given the name because of the colour of wood. Brown rot fungi use simple compounds, of course. They also break down cellulose and hemicelluloses, and that's what they feed off of. Lignin isn't used, and they use this, what's called Fenton chemistry, to get around the lignin matrix, because, of course, the cellulose and hemicelluloses are masked in this lignin matrix. And this is just to illustrate where we find brown rot fungi. This is a wood cell, so perhaps maybe it's a xylem vessel, something like that. The plant cell would originally have just had a cell wall, but then a thin cell wall, but then that thickened with different layers called the S1, the S2, and the S3 layer. Then this is the lumen, the cell lumen. And in the case of those water-conducting cells in the xylem, these cells are dead, but they're filled with water. And the basidiomycetes don't cope well in that sort of environment, but when it dries down, they can cope. And they do grow. And we found the brown rot hyphae in the cell lumen, lying on the wood cell wall, as you can see here. But what it does is, with this mechanism called Fenton chemistry, it makes what are called free radicals in the wood cell wall. Free radicals are little moieties like an oxygen and hydrogen together, OH, or oxygen-free radicals, which are very, very active and can break down cellulose molecules into smaller bits. Now, they don't feed by enzymes. Enzymes can't just get straight into the wood cell wall because they're too big. But these these free radicals that are made in there can break down those big molecules into smaller ones, which then can be used by the fungus. And of course, there's white rot. Now, white rot is a situation where the fungus uses all compounds, all components of the wood, including lignin, and eventually could break down wood completely to carbon dioxide and water. And the white rot hyphae are again sat in the lumen, but this time decay is by enzymes, and these are produced around the hyphae. And what happens is that the wood is gradually eroded, and over time, the whole of the wood cell wall will completely disappear. Of course, things are not that simple. We don't just have white rot and brown rot, we have intermediates. So with regard to white rot, at the two extremes, we have what's called simultaneous rot, when all the components are used at more or less the same rate. And then there's a situation which is called selective delignification, where to start with, at any rate, lignin tends to be used relatively more rapidly than other components. That's not to say that it necessarily does later on. But of course, there are some fungi, illustrated here, that can do both types of decay, perhaps in different regions of the same piece of wood or at different times. And then the final category is soft rot. And in soft rot, cellulose and hemicellulose are used, and simple compounds. Lignin isn't usually broken down. If it is, it's very slow and it's incomplete. And with soft rot, again, the hyphae lie in the lumen where there's no longer any water because the wood's probably dried out, although these fungi can sometimes cope with much wetter conditions. And then they grow, actually put a hypha into the woody cell wall and grow within what's called the S2 layer. And to illustrate how that's done, if you imagine this is the wood cell wall, so this is the cell lumen here, the hypha's lying in there, it puts a fine penetration peg, and it is very fine, about 0.5 micrometers, so on the scale of bacteria almost there, into the woody cell wall. This is the actual woody cell wall here. It branches into a T shape. Enzymes are produced around that T shape to form a diamond cavity. Another fine hyphae will produce probably in both directions, protrude in both directions, and more cavities are formed. And you can see this in these lovely photographs by Mike Hale and Francis Schwarzer. So here we've got a cross section through wood. There's the cell lumen of the vessels, and these here are actually hyphae, or actually cavities, these diamond-shaped cavities within the woody cell wall. That's the woody cell wall. Here we've got them in longitudinal section. You can see they actually spiral round the wood cell wall, and this is because they follow the chains of cellulose in the woody cell wall, which spirals. And here you can see that they've broken through from the inside of the woody cell wall outside. So it looks a bit more like sort of white rot in that circumstance. There are several types of soft rot, type 1 and type 2. You're well familiar with some soft rotters, for example, Kretschmeria deosta, and Ketonium species are often common in wood decay, and many of the xylariaceous fungi. But Basidiomycetes can do white rot too. Indeed, there are quite a few fungi which are predominantly white rotters, which under some circumstances can do soft rot, and some of those are illustrated here. I put the picture of Myripolis in because Myripolis is one such fungus. There are other fungi which are predominantly brown rotters but can also sometimes perform soft rot. And again, there aren't just white rotters and brown rotters. There's a whole spectrum. Those are the extremes of the spectrum. And there are in-betweeners, which we're coming more and more to realise. And these are sometimes termed grey rotters that have some mechanism somewhere between the two. And an example of those would be Schizophyllum communae, illustrated here. Of course, white rot fungi are more common than brown rotters. You've probably noticed this. Brown rot fungi tend to be more common on conifers, but the two examples that I have here of brown rotters, of course, are found on angiosperm trees commonly. So this is the beefsteak fungus fistulina hepatic, which we find on oak and sweet chestnut, and here chicken of the woods, Laetoporus sulphureus, which we get on a range of angiosperms, but also on yew, Taxus baccata. And then just to finish off this section, this illustrates phylogeny of agaricomycete fungi and indicates what their type of decay is. So you'll see within these different groupings, there are many white rotters, but notice this second column, the brown rotters, which have evolved from similar ancestors to the white rotters. And then if we come right over here, you'll notice the ectomycorrhizal fungi, which also evolved from similar lineages to brown rotters and white rotters, sometimes just to brown rotters, sometimes just to white. So just going back to that a minute, sometimes people say, well, you know, brown rotters, oh, they can't decompose lignin, that they're not as good, are they? It's not that they're not as good at all. They have evolved not to break down lignin. Breaking down lignin requires a lot of energy, and effectively a fungus is putting in probably more energy to break down the lignin than it gets back. So the brown rot fungi have evolved ways to get around that by this thing called Fenton chemistry that I've mentioned a couple of times. Okay, things that you already know, but just to get us into the swing of things and to thinking again for next week, where does decay occur? Well, it occurs in twigs and dead attached branches, whether they've got heartwood or whether they haven't got heartwood. Of course, in the heartwood of the trunk, something which I'm really interested in currently, and also in the sapwood, and we should talk about both heartwood and wounds in the sapwood next time. But while I'm mentioning sapwood and heartwood, I'd like to compare the two in relation to their characteristics which allow or don't allow fungi to grow in them. What are the differences between functional sapwood, so functional in conduction of water, and heartwood? Because the thing is, in the standing tree, most decay is within heartwood, if we're talking about the trunk. Yet in a felled or fallen trunk, most rapid decay is in the sapwood. So let's think about this a bit more. So functional sapwood, functional in conduction of water, what do we have? Why is this inimical to fungal growth? Well, for two main reasons. One is that although most cells in sapwood, I guess, are dead, so all these vessels and things that you can see here, the large ones and the smaller ones, they're dead, they're hollow cells, but they're full with water. There are also some living cells in functional sapwood, so in the medullary rays, the ray parenchyma, and these vessels have a living parenchyma cell attached to them. Living cells can defend themselves. Of course, all these dead cells, they can't defend themselves because they're dead. However, they are full with water. High water content means low oxygen and high carbon dioxide. So fungi can't operate in that environment. They need oxygen really for ligninolysis. So there's low nutrient availability. So this is what makes this living cells that are present and this huge high water content in the dead cells makes this inimical to fungal growth. Of course, when the tree falls down and or is felled, those cells, that water content dries out, the cells aren't living anymore, and there's no defense at all, and away the fungi go. So what happens in the heartwood? Well, in the heartwood, the vast majority of cells, possibly all, possibly not quite all, are dead. So there's no living defenses there. But also, they've started to dry out. Those vessels that had conducted water don't conduct water anymore, so they're drying out. So oxygen content is more than in functional sapwood. Carbon dioxide is less, so the fungi can grow. Conditions are great, and they're good from an oxygenation point of view, and the fungus can break down nutrients and can feed. However, of course, in some situations, for example, in the center of oak trees, oak heartwood, has all sorts of inhibitory chemicals, tannins and other polyphenols. Yet despite these inhibitory chemicals, heartwood, because it's got oxygen and not nowhere near the amount of water that there is in functional sapwood, this makes heartwood much easier for fungi to grow in. So that we have to bear all these things in mind when we think about where decay is occurring, in what circumstances decay is occurring in standing trees. So where does decay occur? Carried on. It also occurs in the roots, of course. These roots here were colonized by Myripilus. You can see Myripilus was having a field day. This was also a tree that came down due to Myripilus decay in the roots in this particular case. But there's a brown rotter and a white rotter, and I suspect that neither of these are caused by the Myripilus. There's something else there also, incidentally. Also remember, though, these are woody roots, but fine roots can also be decayed. There's quite a turnover of fine roots, and decomposition of those will occur. Whilst thinking of the tree, I often tend really largely to think about wood, but we should remember leaves and twigs. That decay begins in the canopy, but most of it occurs on the forest floor. And indeed, most wood decay ultimately is completed on the forest floor. So now I'm going to try to answer some questions that were posed to me by some of the panelists about things that people might want to know. And so they're sometimes not necessarily in a particularly logical order, but I hope that they will be helpful to you. So how do fungi arrive? Well, of course, we know they arrive by spores. And I guess there are two main categories of spores. There are the sexual spores and the asexual spores. So sexual spores are produced as a result of sexual reproduction, and genetic material from two parents can be recombined to give other characteristics. That's the benefit of sexual reproduction. And in changing environments, that's very useful. Benefit of asexual reproduction is that the spores will have the same genotype as the parent. And so in a nice stable environment or in a growing season, for example, asexual spores are useful because they have the same genotype. The fungus is growing somewhere, it's doing well, so why fix something that isn't broken, so to speak? Now, so we have sexual spores. These happen to be basidiospores, sexual spores of basidiomycetes. And of course, we know the fruit bodies, the brackets and mushroom-shaped things, etc. Some ascomycetes produce fruit bodies we can see. This is Xylaria, hypoxylon, the candle snuff fungus. This is the sexual state. Now, actually, on those same structures, asexual spores can be produced. You can see that these white ones, this is the asexual state. With other asexual spores, these are conidia, they might just be produced on mycelium. Conidia are the spores made, the asexual spores made by ascomycetes. Basidiomycetes sometimes make asexual spores, or some of them do. They're typically called oidia. But there's another type of asexual spore. These are called chlamydospores. They are thick-walled spores. They're produced actually within hyphae, and they're survival structures. And basidiomycetes produce these as well, I guess, as ascomycetes. And they can be very important with wood decay fungi surviving in harsh environments, like in branches in the canopy. So Buglossoporus quercinus, the rare oak polypore, produces chlamydospores. I think Laetoporus does. Schizopora paradoxa does as well. So that's a mechanism of survival. Now, you might have seen these patterns. I'm sure you have seen these patterns. In this case, this is one of Ted's photographs. I think it was Stereum hirsutum. You can see loads and loads of these interaction zone lines. And if I recall, that the fungi that were fruiting all along here was Stereum hirsutum. Now, when you see lots and lots of interaction zone lines of the same, and the fungus is the same species, we see lots and lots of them, you can be pretty certain that that fungus has arrived by spores when there's lots of different decaycons, lots of different individuals. Here's another example. Sorry about the quantity of this. It was a very old one. The 35mm photo didn't convert well. But you can see, again, lots and lots of individual decay columns. It was Xylaria hypoxylon this time, the ascomycete candle snuff fungus, arriving by spores. But look in this region here. Actually, if you look carefully, you can sort of see zone lines, these interaction zone lines, very faded. This log had been down for nearly 5 years. And this situation here depicts the original picture where Xylaria came as spores, but something has got in here, a basidiomycete, and has replaced all those individuals. That's why you've got this single white rotted region with some relic zone lines in. Carrying on thinking about how they arrive then. If they don't arrive as spores, they arrive as mycelium. This is just a lab tray of soil in the lab, woodblock colonized by Phanerochaete velutina growing out, arriving at this new wood resource as mycelium. Those fungi, or some fungi which can grow out of wood in search of new resources on the forest floor, do so by producing these aggregated structures, these mycelial cords that you can see here. In fact, Phanerochaete does as well. You can see it's making its cords there. You can see the individual hyphae here, and in other regions, it's aggregated to form cords. These cords are well insulated. They can move water and nutrients from one block of wood or one location to another, and they can sit and wait for a long time. This is, I guess, this is just 2.5, 3 or 4 meters. This picture you can see here, but these can be quite huge and quite long-lived. And of course, then there's Armillaria, which produces rhizomorphs. Difference between cords and rhizomorphs is that the cords grow as individual hyphae, which then tend to aggregate, whereas the rhizomorphs, the whole organ grows from the tip. And as you well know, Armillarias can form huge systems in North America. There are many hectares covered by single individuals of Armillaria, and some are thought to be perhaps a couple of 1,000 years old. Okay, carrying on this theme about how fungi spread, think about Heterobasidion. This is a plantation of conifers. The trees have, the roots of the trees have grafted together. So we've got a continuous link between this tree and this tree. Heterobasidion arrives as spores. It could arise on land on a cut surface of a felled or thinned tree and start to colonize the stump. It could arrive on a wound. Perhaps this is a wound made by an animal or mechanical forestry kit. The spores establish, the fungus establishes, but then it can spread from tree to tree through these root grafts without having to grow through soil, which of course is a very dangerous environment in the sense that there are many competitors there. Okay, so the fungus has arrived. And I should say fungi can arrive in different ways. They can be carried to where they're going. They can be carried by animals too. When they arrive, how do they get in? Well, there's lots of different ways. One way is through wounds. These can be large wounds or minute wounds, just naturally occur in small, absolutely minute, less than a millimeter size. They can be above ground or below ground, could be due to human activity such as tree work, could be natural for one reason or another, bending and what have you. Could be due to large animals, could be due to invertebrates. And I've given 2 examples here which cover this, the carriage and inoculation by invertebrates. So these pictures are on the left here, illustrate the situation with Dutch elm disease. The Dutch elm disease fungus Ophiostoma novo ulmi is vectored, is carried by beetles. In Britain, Scolytus scolytus and Scolytus multistriatus. This is the surface of a beetle here. And you can see just on the surface, there are spores. They're carried by the beetles. This would be under the bark. They accidentally pick up the fungus on their outside, like you see here. The beetles fly off. They fly to the twig crotches. They feed in the twig crotches, damage the twig by making a hole effectively through the bark. They eat through the bark and the fungus gets in and away we go. In another situation with the wood wasps, Cyrex wood wasps, here's a wood wasp. This is a mutualistic situation in which the fungus and the wasp have evolved to be so that the wood wasp carries the fungus, actually in special structures. With the Dutch elm disease fungus, it's carried just on the surface, sticks to the surface of the beetles, but with the wood wasp, it's in special structures in the abdomen called mycangia. And when the wood wasp lays its eggs, you can see these are legs here, that tube there, that's the ovipositor tube which sticks into the tree, and through there the eggs of the wood wasp are laid. But at the same time, the fungus, a Mycosterium, is inoculated along with those eggs. The Mycosterium is a wood decay fungus and can colonize the wood, making it softer so that when the eggs hatch out, that the larvae can burrow. And this is a particular problem in quite a few regions of the world, but this example is of exotic pine in South Africa. Okay, fungi can get in through broken limbs, exposed heartwood and sapwood, and I've shown you this picture in many of my talks because I think these are both two beautiful photographs. Chicken of the woods here. And we can see these fungi growing out or fruiting from a wound, and people automatically usually think, oh, that's how that fungus got in. Maybe it did, maybe it didn't. The point is it's coming out of there. Something can get in through there. Doesn't necessarily mean that this did, but broken limbs are nonetheless a way into trees. They can also get in, fungi can also get in by colonizing twigs and effectively being left there when the twig dies. The twig dies, and of course, trees grow outwards as well as upwards, and then the fungus becomes embedded. And a classic example of this is the paint fungus, Echinodontium tinctorium. There you can see it on this tree around these twigs, fruiting around these twigs. So engulfed dead tissues, I've just mentioned. Then there are natural openings, so lenticels, breathing pores in the bark, stomata, the breathing pores in leaves. This is a way that many obligate pathogens get in. And the majority of pathogens, not that we're going to talk about those much today or indeed next week, but the majority of pathogens of plants will get in through intact surfaces. But I would like to show you getting in through natural openings. Now, this happens to be a barley leaf. This is a wonderful image, I think, by the late Nick Reed. It's a barley leaf, so I know it's not a tree, but I think this is really quite intriguing. So it's the underside of a barley leaf. These are the stomata, the breathing holes. You can see that the ridges on the leaf. It's a micrograph, electron micrograph. Now, the spores and the hyphae are actually quite intriguing. They have what is called a thigmotropic response. I say that again, thigmotropic response. That's a response to touch. And you'll notice that there aren't any hyphae growing along the indentations on the leaf surface, because if there was, if that spore germinated there and a hypha grew along there, it's not going to find, or its chance of finding a stoma to get in through is slim. But by growing across the undulations, it increases the chances of finding the stomata. So there's one just going to get that one there. There's this one that's got that one there, because you don't have stomata along every single groove. So I think that's quite intriguing and quite a remarkable photograph. I think is wonderful. Okay, so now I'm coming on to really rather more of a hodgepodge of questions and answers that I've been asked to cover. So why are some fungi selective for certain tree taxa? And I think that the answer to that is they've evolved to cope with specific situations. And this depends on what their lifestyle is, what they feed on, etc. So an example would be those fungi that are heart rot feeders that have evolved to cope with certain extractives. So I put fistulina in again as an example, which we find on oak and sweet chestnut. Over evolutionary time, millions of years, it has evolved to cope with those tannins and other polyphenols in heartwood. And there are not many fungi that can do that. Not many have evolved to do that. So that's one reason for selectivity. Biotrophic pathogens, so those pathogenic fungi, which can only invade living cells, at least initially, have evolved to cope with some of the defense mechanisms of a specific plant species often, even a specific strain of a plant. And then another example might be, and again, we'll talk about this next week, those fungi which are latently present in functional sapwood and which then become the first colonizers when the branches or trunk starts to dry. Different tree species have different groupings of fungi that are the first colonizers developing from these latent propagules. And yet there's a whole soup of different propagules in the wood. And the thing that makes them selective, in inverted commas, is the conditions that occur when the wood becomes dysfunctional. In other words, how quickly does the wood dry, and other features like that. Now, there's another thing that I want to discriminate to you, actually based on a talk that I gave earlier in the year. Somebody said in the questions that when we're talking about these interaction zone lines, so these lines are caused by fungi. Somebody said that they thought that that was the tree and that it was a codit defense. Now, these interaction zone lines you can see here, they are demarcations between and made by fungi. Can be fungi of different species or it can be fungi of the same species. This orange one, this is Mucidula mucida, we used to be Eudermantiella mucida, and that's very often, that's the most common orange one that you can find. So these are all interaction zone lines between different individuals, as I say, can be the same species. Same species in this case, I think these were mostly Sterum hirsutum. There are different species here. These lines here, this is Kretschmeri deoster, and you can see that the individual that is there has produced its zone line, and the individual that's here, it's produced its one too. So that is distinctly a double zone line there, and you can see the no man's land between. Intriguingly, when you get no man's lands between interaction zone lines, you get some microfungi that can manage to eke out a living in that inhospitable environment. I just wanted to point out that these patterns here, this is partly due to the tree, partly due to the fungus, that the white areas are where they're more decayed. This is typical rots caused by hypoxylon species. Okay, so those interaction zone lines are fungi that fight each other. They're surrounding the fungus territory in the way we have fences or hedges around our homes and gardens. But remember, fungi attack each other, and I couldn't resist telling you a little bit more about attack, only on one slide, because this could be a whole talk in itself, but just to remind you that fungal communities often change because of these interactions between mycelia. Fungi can interact at a distance. You can see that here in agar culture. There's a fungus there. They never manage to get to touch each other even because of diffusible chemicals in the agar or volatiles in the air. Then there's hyphal interference. That's illustrated here. This is Phlebia gigantea, that hypha. This is a Heterobasidion species where Phlebia has touched Heterobasidion. You can see the contents of the fungal cells are in a terrible state. Phlebia gigantea has killed Heterobasidion, and that's the basis for the biological control of that fungus. And I suppose this is a simple form of mycoparasitism, contact mycoparasitism. Another mycoparasitism is illustrated here. These actually aren't wood decay fungi, but the images are so beautiful. Thanks to Peter Jeffries, you can see this hypha completely in very tight contact with this one. And there's a structure, an apressorium, and from there, underneath there is a hypha which penetrates into the hypha of the other fungus. And then there's this main category that I've worked on so much where the whole mycelia fight on a massive front, and we've called that gross mycelial contact. There'll be lots of different mechanisms. We just know what they are. They'll be categorized by people long after I finish doing research, I'm sure. So here we've got Phanerochaete velutina, which has started making cords. Look, there's its mycelium before it's aggregated, attacking Hyphiloma fasciculare and gradually pushing it back, probably producing enzymes, all sorts of chemicals. And this fungus will replace that one. Okay, so those interaction zone lines are also sometimes called pseudosclerotial plates and typically demarcate territory occupied by different individuals when they're fighting each other. But there are some fungi which produce these pseudosclerotial plates, apparently in the absence of other fungi, or at least they produce them very much more than some other fungi do. These are plates of fungal tissue which have been melanized, they're thick-walled, and they maintain the conditions within the wood such that it suits the fungus that's made them. This is armillaria that's doing this, and people often misidentify this and think that it's wood that's been charred. It's armillaria again, broken open here just to show you that soggy, typical armillaria wood. So with armillaria makes pseudosclerotial plates around itself to maintain the wood in a wet state. And that actually protects it quite a lot from attack, I guess, from other fungi that can't cope with those high water content conditions. Other fungi like Xylaria, Hypoxylon, candle snuff fungus also produces these pseudosclerotial plates, but for a different reason. That is to keep the wood dry. Ascomycetes cope better in dry conditions. You'll see Xylaria sometimes encased in these black pseudosclerotial plates. The wood is very, very dry. Xylaria decays it very slowly because it's very dry. That's its strategy. And that's probably a mechanism, again, for keeping other things out because it's so dry. So at one extreme, so wet, another extreme, so dry. Here we've got a few other examples of features we see in wood. These are obviously interaction zone lines, but I wanted to point out this sort of staining here. This has been cut. This is dark colored. This is dark colored here because it's wet. Cut through these pseudosclerotial plates and the water flowed across. This was white, this white color like this before these were cut, and the water has flowed straight in and made it look darker. And talking about water, we often see this condition in wood, wet wood. In this case, the wood, this is in beech, and I think all the examples we found places where the water would get in higher up the trunk. And when the water's got in, it's made those central tissues wet again, even though they started drying out. Fungi might have been there originally. You can often see the patterns where the fungus was. But now, because it's so wet, that's inimical to fungal growth. And we have bacteria in that wet wood. There are some other conditions in trees, for example, willow, called bacterial wet wood, which is a pathological condition caused by the bacteria. Okay, a few other tree features before I finish. Sorry again about the quality again. These were from 35mm slides. I just couldn't lay my hands on better pictures at the time. So you will have seen these things. This is an oak branch. You can see these wings of heartwood. There's a winged collar and these wings of heartwood. This is heartwood and there are wings sticking out. This is the situation before the sapwood's gone. Heartwood in the middle of this big branch, and here are these wings, heartwood wings. Those are produced by the tree. They are prematurely formed heartwood. Then the sapwood rots away. Eventually that would also, and you get to this situation. Section through beech here. You can see all the interaction zone lines, lots of Udomantiella again. But this is possibly tree-induced. Might be called a reaction zone using Shane's terminology. And we'll talk about that in connection with wounding next week. These are also features that we might see in trees. These dark lines, this is in beech, this is what's called red heart. I know it doesn't look very red now because it's well decayed, but these dark coloured lines, these are, this is wood, this is rather differently constructed wood, probably, and all the light coloured because there's a fungus there now. So I'm sort of imagining that maybe in this region here, the fungus managed to get in and started to decay these central tissues. And it was all sapwood here at the time, but there were some living cells which could make this different wood. And then over time, it gradually moved out and this process continued. There's another thick region of wood there, much more lignified, I guess. And this is my final slide, which really is, I suppose, an advert for next week. When Alan and I wrote the 1988 Wood Decay book, we thought that there were probably about 5 main categories of ways in which fungi colonize trees. And I think it probably still works now. There are the heart rots, and I'll talk about those next time. There are those which are pathogens, not talking about pathogens. There are those which we call unspecialized opportunists and specialized opportunists. In other words, wound colonizers. We'll talk about wounds next time. Those are the unspecialized ones, the fungi that just get there because there's a wound and they just happen to be there. And then there's latency, those fungi which are present in functional sapwood and established from there. And then, of course, there are those fungi which get into trees not at the start of decay, but they get in and fight fungi that are already there to gain territory, secondary colonizers. And I don't think we'll have time for that next time. So next time, heart rots, wound colonizers, latency. So on that note, I'd like to thank everybody who over the years has contributed directly to this research, which I've talked a little bit about today. And thanks to everybody who supplied me with wonderful photographs. And thank you guys for listening.
Great stuff as ever. Thank you very much, Lynn. Right, well, look, there's a panel of excited people in front of me. Ted, just to start off with, have you got any particular thoughts you want to cast off with? We've got plenty of questions too to work through. You're on mute at the moment though, Ted. I wisely muted you. It's the best thing about Zoom webinars. Although now I'm regretting it because this is going to require some technological input. While Ted's looking for the mute button, Caroline, Mike, Jill, anyone like to chip in with anything to sort of lead the way on what Lynn's just talked about? Well, Ted's on now, look, typically.
Right. I think the one thing to remember is that these fungi co-evolved with the trees. And they're not just bad news. I think that is one of the things we've got to stress, that we know now that they're not bad news. They co-evolve with the tree. So, and hopefully Lynn is trying to tell you about the mechanisms which they enter the tree, but it doesn't mean to say they're going to kill the tree. And we've heard about how, I mean, they compete with each other, and everything has another thing that eats it. Whatever you remember that. Parasites have parasites have parasites. So it's a co-evolutionary relationship. Thanks, Lynn.
Of course, that's very, very important for pathogens and things. I mean, we have lots of problems with pathogens because we move them around the planet, and be it tree pathogens, other plant pathogens, pathogens of animals, frogs and toads, or what have you. What we typically find is that in the region where the fungus co-evolved with the plant or animal, there's no problem. And that's true with the ash dieback fungus, with Dutch elm disease, and so on and so forth. So we are the major problem causers.
Go on, Jill.
Sure.
So yes, I mean, a lot of D words, decay, a lot of R words, rot and stuff. And I agree with Ted. I mean, it almost seems like, how do plants survive the onslaught particularly? But I'm interested in the fact that you could say, well, very few trees get through to the ancient life stage, and we've got very tiny proportion of ancient trees. So maybe they fall by the wayside and they're just exceptional. But actually, I'm starting to think about collections of old trees like Windsor, like Sherwood, Savanac. There, you've got a lot of old trees and a whole age structure And I was very struck at Duncombe when there was a study at Duncombe. Some of the biggest trees had just a tiny bit of the fungus in it, but but what I'm thinking then is, and the other thing is, of course, we've got all these machines to be able to tell where there's decay, but actually, it strikes me even more strongly, we need something that tells us about sapwood and how much water there's in the sapwood. Surely there's a machine that can look at water content and tell us whether the thing is repulsing the decayed fungi. Just a few thoughts.
Yeah, I think from the plant pathogen point of view, I think most plants are, well, immune's not the right word, but immune to most pathogens most of the time. I mean, we tend to have it, when we have an issue, we notice it. So we notice it with our crops because we grow them as vast monocultures, and a fungus will have evolved to cope with the defense mechanisms of that particular plant and will swamp through. So we see, because of the way we grow things nowadays, we see this devastation by a fungus, but that's not really usually the natural situation at all. In the natural situation with plants, not necessarily trees, but other plants dotted about here and there, you don't get these problems usually with plant pathogens. And indeed, when you do have little patches of pathogens, it increases diversity. So lots of the problems we have are problems we've made ourselves from a pathogen point of view. Most of the decay, so not with pathogens, most of the decay we get in wood is not a bad thing for the tree at all. Mostly it's a good thing, although I'm a little bit biased on that, obviously. I should say that there are some fungi, like, for example, Heterobasidion, which foresters consider as pathogens. I mean, I think that it can kill living cells, so technically is a pathogen. But I think most of what it does is in the middle of a tree. And those trees will live for a very long time. They're probably not being killed by Heterobasidion at all. But the foresters think of it as a pathogen because it's a nuisance and they can't sell their trees for timber to do whatever because there's something in the middle. And the whole reason why in Germany they wanted to look at heart rot fungi originally because of all their beech trees. So we shouldn't really usually most of the time be thinking of decay as a bad thing. It's an essential process.
It's also about, sorry, Lynn, it's also about monocultures and the way we grow these trees to try and maximise on profit. But actually, diversity is better management of those situations.
Yeah.
Well, okay, then I'll have another go. I think one of the things is in my travels across the world, you realise how advanced and experienced arborists are in the UK about old trees. And so many of them actually learned climbing about in old trees, whereas in many other countries, they don't have them. So their perception of trees are, if you like, lollipops, whatever. They don't have the number of old trees we have to see, if you like, what I would call natural decay processes, which aren't necessarily caused by the fungus or the fungi, but are actually just natural aging. We know that most trees, it's only the living tissue which is the real tree, especially with the true heartwood species. So therefore, we have this wealth of experience which we can pass on because we're that far ahead in our observations on just what happens inside a tree, the hollowing. You showed a lovely beech tree on the ground, nearly your last picture, which showed a foul tree with all the gunge coming out. But there was a distinct line. It was distinct, and it wasn't going into that living tissue on the other side. Okay, what caused that? What was that doing? And so I think, depending on the mushroom, then we've got to think is what actually is it doing and will it change and will it adapt? But we have the experience because we have so many old trees, and hopefully you're going to get some questions from this audience.
Yeah, so trees, I mean, they have a huge amount of protection. They have protection around the outside, preformed barriers in the form of bark and chemicals and what have you. But the major protection, as far as I'm concerned, in functional sapwood is the fact that there's this high water content, which they have to maintain because they are shooting water up from their roots to their leaves. And I think that in itself is just a major protection from ingress by any decay agent. But in the central regions of trees, where the tissues are not only dead, but don't contain very much water or contain a lot less water and certainly a reasonable amount of oxygen, that's where fungi can grow. And those tissues are not functional in water conduction anyway. One thing that really winds me up, really, and it's in the scientific literature as well, is when fungi are classified as being tree pathogens because they've been found fruiting on a standing tree. And that, in most cases, is absolute rubbish. The fungus is not being pathogenic. It probably was never pathogenic on any part of the tree. It's there feeding on dead tissues. It is not a pathogen. Now, there are some fungi which feed on dead tissues, which can get in by being pathogenic. So Heterobasidium, I think, is probably an example of that. And there are other fungi which get out by being a bit pathogenic, by killing some of the living cells in sapwood to get out, to fruit outwards. But most of the fungi which we see most of the time on trees are using dead tissues. They are not killers, in my opinion.
Caroline, if I could possibly bring you in here as well, because we spoke earlier on and we chatted about the conference in 1993, the first sort of Talking Rot event that you were involved in, and some of our other panelists were involved in as well. And you said that it was quite frustrating that back in 93, we as an industry were saying exactly what Lynne's just said, and no one seemed to listen. And here we are talking about it now. Are we moving in the right direction, do you think? Is there greater understanding of this?
Yes, there is greater understanding, but I think some of the difficulty is some of the complexity that Lynne has described. In other words, we can't know and see precisely, or even in a macro sense, what is actually going on within the tree. And although some of the equipment we have enables us, possibly, if we can interpret it correctly, and the equipment is actually telling us what we're told it's telling us, it can give us some indication of the effects of the fungi on essentially wood strength and its distribution of fungi within the tree by its effects. Our difficulty in many situations is, well, what does that mean in terms of the usually the safety of the tree? In other words, what strength the tree has, and progress of that. And the problem is we don't go back and do enough follow-up to know, or we don't have long-term, and they would have to be very long-term experiments to know the kind of progress of these things, because there is the tendency, when a certain amount of decay is found in a significant tree in a location where it has targets, that the tree is felled rather than it is monitored in some way. And I think we're still missing some of the equipment to do that in a way which people, I think, feel confident about. So we still do that. And I have to also say, if I mention the T over R thing, if everybody knows what that means, that you can lose a certain amount of sound wood or strength from a tree before the tree actually has any real impact on its ability to stay standing. And that has been in a way misused and unfortunately in a not very understanding way, to actually say that some trees are unsafe when clearly they're not. And one of the things about Ted was saying in relation to what we're learning from studying ancient and veteran trees is you have to ask yourself, you saw the opening slide Lynne had of the tree with a very, very small amount of live wood on the outside and the entire centre colonised by a whole array of individuals, we don't know precisely what. But the tree, until such time as it was cut down, was standing on that very small amount of live wood. And we've got to try and refine, our understanding of how much strength a tree requires to stand. And the bigger trees, the bigger tree gets, that also affects the actual diameter of the tree, affects the strength and the ability for the tree to resist breakage. So all of those things, we've gained some knowledge, but there's still lots more we need, I think, to make decisions. Otherwise, people fall back on, it's always, you're safer to fell a tree than to keep it up if it's got sort of decay in it. So we're still in that position.
Can I jump in there?
I was just about to ask you to jump in there, Mike. I should have thought you right. Go for it.
Before you do, there's a question just flagged up which says most hollow trees break. So that is a perception of people out there. And I guarantee she's not English or British. She's not coming from the UK where we do have hollow trees. Sorry, Mike.
Just really to tie together something that Lynne was talking about and what Caroline has said. If we are prepared to acknowledge as an industry that functional sapwood generally isn't decayed by decay fungi, then we need to develop an understanding of sapwood distribution across the range of species that we deal with on a regular basis. So we have a mature, healthy tilia, has a very high proportion of its cross-section with living cells. It's functional sapwood, high moisture content, very limited capacity for fungi to degrade it, whereas a mature or perhaps even post-mature oak will have a very thin wall of functional sapwood. So different species, at different ages, different stages in their lives, colonized by fungi in different ways, or certainly volumes of fungi vary very much. And we can do it. Caroline was talking about equipment to investigate decay distribution, functional, structural tissue distribution. But I think we can do a lot of that without that equipment, just an improved understanding of the way that trees lay down their tissues and the way that those tissues are subsequently degraded by fungi. I've just got one note from right at the beginning of Lynne's talk, where she was talking about, Lynne, where you were talking about saprotrophs, necrotrophs, biotrophs, and beneficial fungi, that some fungi are beneficial. And my thought immediately was, aren't all fungi beneficial at some level?
Yeah, obviously they are in terms of whole ecosystem functioning. I think that it's a human construct, isn't it, whether things are beneficial or not, or else you have to define who benefits whom or whatever. So if you're thinking in the sense of a tree, then one might not think that a necrotrophic fungus is beneficial to it if it is killing it. But if you're thinking about the whole ecosystem, it could be very beneficial to the ecosystem. Certainly if it's plants dotted around here and there, an increase in diversity. Yes, so those sort of categorizations really are a little bit dangerous, I guess.
Yeah, and the interrelationships between fungi and invertebrates or the fauna and trees are very, very complex. And I always remember at one of the first Ancient Tree Forum meetings up at Duncan Park in North Yorkshire, and there was a debate going on that Caroline was quite heavily involved in at the time, I think, about the aesthetics of leaving standing dead trees, particularly large standing dead trees. And the Keith Alexander was one of the group at that meeting. And Keith disappeared for quite a long time, and when he came back, he was all excited because he'd found a crabapple. And this one crabapple was the habitat for part of a life cycle of the beetle that he'd found earlier on an oak tree. And there was only the one crabapple within the travel distance of this particular beetle. So very, very, very complex relationship. So without the crabapple, that beetle would have been lost from that site.
Yeah, I was actually going to talk about standing dead trees. To me, they are very aesthetically pleasing. I like living trees and I like dead trees. They're very important, standing dead trees, as habitat for other organisms, be they fungi or invertebrates or combinations of both. And indeed, we're probably losing a lot of fungal diversity because we don't have very many standing dead trees. I mean, I think the classic examples probably are the kilo trees in Fennoscandia, these pines, which I don't know, they live for 500 or 600 years, and then they'll stand dead for another 500 years. And that habitat which they create, there's no other habitat like it. And so, of course, you get organisms which specialize in that habitat. But and the same is true for our standing, whatever standing trees we have, we see them so beautifully standing in fields sometimes. They're very special habitat, and they really, in my mind, should be protected to the same extent as standing living trees.
Thank you.
Can I say amen to that? As you know, our laws don't protect dead trees. Even though our policies, England policies do protect them. And it would be very nice if also the management of dead standing trees were not as severe as I so often see now. And again, this is an excess of caution. It removes so much of the habitat which could endure for so long on these dead standing trees. Clearly, you have to take into account the durability of your species and other factors relating to the inevitable decay of the root system. But certainly with long, durable trees, we could, I'm sure, leave rather more of these trees than currently I see. Could I ask Lynne, particularly, to address a question which was posted by Ian Hanna because of our ancient and veteran tree interest? And he asks, is it sufficient to conserve host trees, such as ancient and veteran trees, to support rare fungi, or are other measures equivalent to seed banks or translocating or reintroductions? Could I just say that we keep ancient and veteran trees, or they're important not just for rare fungi, but because the whole habitat of decaying wood is a rare habitat. And these trees are the source of that habitat and all the associates that go with it. But could you think about that question, Lynne?
Yeah, so I support you very strongly in what the last point that you've just made, Caroline, about the importance of habitat for other organisms. I think that probably hollowing trees, the habitat. Yes, okay. As a mycologist, I'm interested in the fungi, and they're a rare taxa, which we only really find in those circumstances. I think like the rare oak polypore, things like that. There are also rare invertebrates. So, for example, violet click beetle on beech and ash, at Windsor on beech, and a few other places. Well, actually, near where you and I live, Caroline, on ash. But many people are not interested, I guess, in invertebrates. They're not interested in fungi. But straight away, if one tells them that probably worldwide there are something like, I don't know, 1,500 species of vertebrates which are dependent on hollow tree habitat, I think that is something which perhaps captures the imagination of many people. In the UK alone, I think there's something like 1,700 species of invertebrates dependent on that habitat. So it's hugely, hugely important from a wide perspective of different organisms. I've forgotten the rest of the question now.
Sorry, the question was, can we we can keep the trees because they have these fungi. Are there other measures equivalent to seed banks or translocate or reintroduction? In other words, can we artificially create or conserve these fungi? But I would just say not just rare fungi, the fungi associated with that habitat, perhaps that question.
Yes, that's a good question. Does Ted want to add something to that before I answer?
Well, we've actually been doing that at Windsor now for the last 10 years. And with Pete Wells, an ancient tree forum people, but then also the horticultural department at Windsor, where we've been grafting scions from the old trees onto saplings. And so we're getting a gene bank. We're getting a gene bank of the ancient tree. But then it was pointed out that when we go to plant them out as saplings or whatever, standards or whatever you're going to do, some of them ought to go near the ancient tree in the hope that the soil community, especially the mycorrhizal fungi, transfer from the ancient oak onto the sapling. So then we get what we are now calling biological continuity. This is what we hope we're going to get. We certainly will get the gene banks. And one of the interesting trees there is the original done was the Pymphadic oak from Wales, their biggest tree, which blew over. Murray managed to get some cuttings. They were grown at Windsor and they were grown by Pete Wells. And what's happened with that one? One of the saplings now is 2 to 3 meters tall. One of the grafts, I mean, and it's actually producing acorns because the sapling is still behaving like a branch. It's fantastic, but it's going out near other, in this case, by ancient oaks. But hopefully one where we go back down to the Pymphadic oak thing. So there are moves. But I think the two moves are saving the gene bank, but also then the soil community, because let's face it, half the tree is under the ground anyway. So we're going to get biological continuity of a couple of thousand years.
So, sorry.
Can I just jump in there? Just talking about continuity, there's quite a bit of the chat is around ash dieback, and it seems to me that one of the things that we need to be mindful of with our management of ash dieback is not just the management of the genetic material in the ash tree itself, but also its associates. So retaining as many dead, declining, what we might perceive as being lower quality ash trees is essential. I recognise that there are significant financial constraints, financial burdens, particularly on large organisations with lots of ash. But nevertheless, retaining as many ash trees as possible in whatever condition can only be a good thing.
Yeah, well, I'd absolutely agree with that from external habitat and internal habitat for lichens and what have you. But just to answer, I think what Caroline may have been asking was, is there something equivalent to seed banks, and can we do things with fungi to preserve them in association with trees? So you can store fungi, you can keep a living culture collection. And we have our own small living culture collection, but it takes a lot of upkeep. But there are places like Kew and the Vesterdijk Institute, which have living culture collections. But yes, we can inoculate trees. I'm hoping that that's what you're asking, but even if it isn't, it's what I'm going to say. So we do have a project or set of projects looking at inoculating trees with threatened fungal taxa. So in particular, we've been trying to inoculate Hericium species, these tooth fungi, these pendulous tooth fungi, Hericium erinaceus and Hericium coralloides. And we are actually, obviously, it's a long-term thing. It's a legacy experiment. You set it up now, and I won't be here probably to come back and check on it in however many years' time, 30 years or whatever time. But just so we could just get a clue as to whether we're having any success after 2 or 3 years, we've just started sampling. And yes, we have managed to inoculate by boring holes into the tree and inserting colonized dowels. We have managed to get hericiums established in young-ish beech trees. And then another part of the same thing, so that's that's conservation inoculation. And then, of course, lots of people will know about veteranization of young trees to try and to get veteran and ancient trees rather more quickly, because we've got a big gap in the cohorts from all this massive planting to the wonderful veterans and ancients that we have. So rather than just following the idea of veteranizing by tearing off limbs or, I don't know, setting fire to the base or whatever, some of the approaches that, very good approaches that Vicky Bengtson and other people like that have done, we've tried to be a little bit more targeted, and we have a couple of sites, one of which is Windsor, which Ted has helped us with tremendously, where we are actually not just making holes in trees and hope that hollows form, but we've made holes in trees and hope that we can inoculate those heart rot fungi, which we think might be important. And I have to say that, things are looking pretty good, pretty promising from our first look-see. Had a quick look a couple of weeks ago, and things are very good. And we're now sampling those to see what fungi have established, either things that we've inoculated and or things which we didn't inoculate, but which may find their way in through the holes that we've made. I don't know if that's what you were asking, but that's a story for another day.
That was the kind of question. I would just say that we need to just exercise a degree of caution here, because the dimension that we need to consider is time. And, this all to create the kind of habitat that we associate with a veteran tree, a tree which we are trying to conserve because of the entirety of its habitat, is a factor of time. And whilst it's also very good in conservation terms and bridging gaps and that kind of thing, and increasing the amount of this deficient habitat, that doesn't mean to say that it isn't a substitute for those trees which already have these habitats and are currently fulfilling a very important role, because, as I said before, we generally have very little of it. And like ancient woodlands, the percentage of ancient woodlands is very small. The numbers of trees that actually have this kind of habitat. I mean, unfortunately, arboriculturalists, it's a bit like policemen and criminals. Arboriculturalists tend to come up against a lot of trees which have some of this habitat. But if you think of it in the bigger picture, then these trees are not common, not in the condition I'm talking about. Clearly, decay is a common feature of trees, but the overall veteran condition with reasonably established and advanced decay, with decay and hollowing, is not a common thing. And certainly standing dead trees are not common. And if we are to retain the whole community of organisms associated with them, then we need to conserve what we have whilst trying to ensure we can perpetuate that, those kind of trees.
I agree 100%. Very well said. Absolutely.
There's some great questions coming up on the Q&A, and I'm not quite sure how we're going to deal with them in the time frame. But there's one there that struck me. It was, how do we help trees fight decay? Now, we've been talking about how decay is important in the whole system, but so I might not phrase it with the word fight, but there's lots that we can do to make it better for trees, that they don't get catastrophically precipitated into decaying. Any thoughts on that, Lynne, or anybody else?
Yes, please, yes. I'll return the question to whoever it is and say, can she explain why we have ancient trees? Why do we have trees that last 1,000 years?
Yes, Ted, but what
meaning that it's she's missed the whole point.
Well,
Okay, if our trees are healthy, the sapwood is fully functional, then it probably won't decay. Basically, decay is not a bad thing. The implication of the way, I'm not sure how the question was written, but the sort of the way that you said it, Jill, made it sound that decay is a bad thing, and decay is something that is invading the tree. That really isn't true. And I know you know that isn't true, but it's sort of the way it came over to me. It's the fungus is in there in circumstances where that tree is not functional in doing its water conduction, or that part of the tree where you find the fungus. The fungus will only be where it can be. It's unless it's a pathogen, and there are not very many of those really. Unless it's a pathogen, it can only be where the conditions are okay for it, and it's not where the tree is doing its tree-ish things.
Can I add to that? That's the whole point. As arboriculturalists, we need to be focusing not on the decay. It's actually on the health. Okay, what is a healthy tree? We've said a healthy tree is a tree that has fungi as part of it. But the health of the tree, i.e., what are the things that actually tip the balance? Everything is in a dynamic flux in the world and in the world of trees. But we know the kind of things that actually we want to remove stressors. And the big one is drought and damage. And of course, our activities of pruning cause damage and cause drying. And those things, and aeration, and those things change the whole dynamic of the fungal communities within trees and permit extension and other colonization and change which things are in the ascendancy in a tree. So we've got to be looking, and it's not, I'm afraid, it's not just things that we have control over, because there's aerial pollution and obviously droughts and stress that we don't have direct control over. But where we're looking after trees and we're trying to ensure, if we see the kind of stressors that cause trees problems, we should be trying to alleviate those. And the tree will then manage its own fungal community, if you see what I mean, in a way which keeps in balance between its growing and its decaying.
Oh, Mike.
Yeah, well, I like part of what Caroline said there, because very often we prune trees, we identify decay in a tree, we see the decay as being potentially a structural weakness or resulting in structural weakness, and then we seek to reduce load by pruning the tree. But often that's the worst thing we can possibly do, because as soon as we start removing foliage from a tree, we remove vascular function. If you remove vascular function, you're killing sapwood, and the decay extends. So it's possibly a much more negative contribution to the structural stability of the tree in many situations is to prune.
Well, as I say, where we have to prune, I think we have tended again to be too hard, too conservative in our judgments. I know it's difficult. We have all the issues to do with safety, and we don't. Again, can we calculate these things with great difficulty? So particularly, we've always said this with ancient and veteran trees, is that if there is pruning to be done, it should be done, the least possible. And we must remember that load removal of end weight, it's not a linear relationship. It's what am I trying to say?
Volume.
No, the leverage. You can remove a small amount of length of branch and get a great reduction in leverage. Sorry, exponential. And therefore, you should try and minimize the amount of reduction of photosynthetic area or branch leverage. And if at all possible, spread it over time. And that's something, again, lots of things are difficult to do in the real world. But these are the things that we should be trying to advise, because if we're trying to tell somebody how we want to keep this tree alive and healthy and long-lived, we shouldn't be compromising it by activities which we claim to be assisting it. Difficult, I know.
So I also think we shouldn't be starving trees. I know that, in the past, we've starved trees terribly by removing material under the tree. The annual leaf fall is so important for the system. I mean, we've got to have that leaf litter back there on an annual basis to feed the root system. And we've got to look after the soils, of course, and we're under pressure because nitrates in the air are affecting the soils, and we can't do much about the nitrates, but we can perhaps make sure that the soil system is as effective as possible. And from regenerative agriculture, we've focused too much on single species under trees like grass. And we know that grass is terrible because it doesn't have a diversity of root structure. So we need to diversify where we can, and it's not too shady, the range of plants under trees so that we're improving getting the humus back into the soil and getting all the effectiveness. But so it's also about that, and beyond the drip zone of the crown, out further than that if we possibly can.
I've got a bit of an anecdote there. I think it was 2000 to 2006. We ran a workshop at Silwood Park, 3-day tree safety management workshop. And every Saturday afternoon, it was a Friday, Saturday, Sunday, and every Saturday afternoon we went out to Windsor, to the park. And the first year we turned up in the car park, and there were people picnicking underneath the old veteran oak trees around the car park. And not only picnicking, but they sort of all nosed their cars into the trees as well underneath the canopies. And all the trees around the car park have been dead wooded, all the old oak trees have the dead wood taken out of them. So quite a debate followed, and we suggested that they stop cutting the grass underneath the trees. And Bill Cathcart, who was the park superintendent at the time, did exactly that. He stopped cutting the grass. They later put knee rails up around the car park so cars couldn't get onto the grass at all. But with the gang mowers not going underneath the trees, they didn't need to keep crown lifting them. I went back 2 years ago, and that was after 19 years, and the canopies of the trees were down within a metre of the ground, and that gap was filled by other vegetation. So all of the foliage that falls from the trees, all of the deadwood that falls from the trees, the whole ecology, the whole soil ecology of those trees has changed quite dramatically over that period, and they look pretty good.
Have we got any more questions from the audience?
Have we got any more questions from the audience? Ted, we've had about 40 questions in. I think we've done 2 of them. We're already 10 minutes later than I promised we'd stop, and it's all brilliant.
There was a really interesting question that relates to some of the work that Lynne has been doing with Emma, or was doing with Emma Gilmartin. And it was, do you think that the host species for fungi or the host fungus relationships are changing?
Yes.
Yeah, I think there is evidence of that. I mean, we think that there's evidence for those fungi. Well, I know that there's evidence for all sorts of things changing. Some things we understand perhaps, or have a slight handle on. Other things we haven't got a clue about. So, for example, of fungi which are latently present and then are the first things to emerge, with the ear fungus Auricularia auriculojudae, which in the past we've typically almost always seen on elder. that now is appearing. I always use this example, and it'll come back to bite me one day, and we'll find out that it isn't latently invading fungus, present fungus at all. But it isn't predominantly on elder nowadays. It's on lots of different tree species. And my hypothesis at the moment would be that's because it's probably latently present. And because of climate change, conditions are changing when branches become non-functional in conduction of water. So the rates at which they dry and other features are changing, the temperatures are changing, and that selects for different species from that soup. So that's the hypothesis I'm working on. Time will only tell if that's right. But there are lots of examples. I'm working with Alan Gange from Royal Holloway College at the moment on a manuscript about ectomycorrhizal fungi, and it seems that there's been a change in ectomycorrhizal species that you find between oak and beech. And I'm not sure that we know for why, but I think the point is that there are things which seem to be changing, and it's likely related to climate change or other aspects of global change.
Mm-hmm. Well, I've got quite a good example, and that was in the early days looking at brackets on mature trees, and this was internationally, this was Europe, a thing called Ganoderma resinaceum. I only ever found it on beech, and today, David Humphreys was pointing this out, it's become a species on oak. Now, I still like this talking about oak has true heartwood and beech has ripewood. So you've got two systems, and this thing has moved over to, if you like, to what I would call a much more hostile environment. This is resinaceum. I mean, I only have one tree which has resinaceum on it now, but everybody will point out that resinaceum is actually on air oaks with true heartwood, but also you find it on turkey oak, which has a similar ripewood to beech. Tell me about it. Are we looking at two species, two races? What's happened? But they have definitely changed in the 40, 50 years I've been dealing with brackets.
Yeah, it's something we can touch upon again, I think, next week, probably. I think if we had clues as to what all the changes are, if we made a list of all the species which seem to be changing, we could then interrogate data sets, because, of course, people have been recording fruiting of fungi for a long time, only because they're interested in it with no particular aim. But it turns out that it's really important for seeing changes, and we're investigating this for climate change. So if people wanted to provide a list of things that they think are changing species, we could investigate that. But of course, one thing we have to be cautious with is that fruit body is an indicator of a presence of a fungus, but the lack of a fruit body doesn't mean the fungus isn't there. And when I talk next week and I talk about what we found in beech heart rot, you will see that very clearly. And it's a very different picture in oak. So I think, yes, things are changing. We could start to investigate this. It would be a great thing to do if we had a list of candidate species, if you like, to look at. We could do it across Europe because we have data sets accumulated from across Europe in the consortium I belong to. But I just put in the caveat, the warning about it doesn't mean it wasn't there. In the future, I think we'll be able to look at these things. It won't be me again. In a long time hence, we will have a better idea, because we'll have been able to find out what we have in our trees using other methods than fruit bodies, so perhaps molecular methods or what have you. And we know that those things, what things are there that aren't fruiting, and we'll be able to compare. We haven't got the luxury of that at the moment. We have to make do with what we've got, which is fruit bodies, but those records may not be telling the whole story.
Even with your molecular studies, Lynne, they may be there, but what are they doing?
Yes, well, that is a very good question, and it's a question that in one of the lectures this morning I was posing that same thing.
It gets us so far, but only so far.
Absolutely, these molecular techniques are very sensitive. They detect bits of DNA of maybe spores of dead fungi, of bits of who knows what. That probably many of them may not be playing an important role at all. So yeah, molecular tools are not the panacea of all tree biology.
There's a question just come up about cutting branches off, and so they can occlude, meaning they can seal over, because presumably that person that's put that question up is assuming that decay is bad news. And I think we got Mike, we got Caroline, we should be talking about not seeing decay as bad news. And their perception is that a solid tree is a healthy tree. This is 2021, not when I first started in the 60s. So we've got to get across somehow, especially to arborists, that solid wood doesn't necessarily mean the tree is healthy and the tree is safe. So that is why we're here. That is what we're all about. But 20 years on and hearing that really...
Next week.
Yeah, absolutely.
Well,
next week when we talk about wounding and things, we could pursue this.
What a beautiful segue to next week. I knew we'd find a perfect segue to next week, and I think we found it. We're going to have to draw it to a close just because you've all got busy social lives, I'm sure. Not you lot, obviously. I know you haven't. I mean, all the people out there watching it.
I can't sleep.
Of course you have, Ted. I know you're going out. I know you're out any minute. Look, that was amazing. I love that. I've never felt more redundant, and I've never felt more delighted to be redundant. It was absolutely great. Brilliant. Thank you all so much. I think you all enjoyed that out there too. We have got loads of questions. There's 37 open questions. We've never finished with that many. What we're going to do, as always, we'll make sure all the questions and all the chat are sent over to our panel. We'll probably try and do it tomorrow. And then because we've got the luxury of another week doing it tomorrow, we'll probably be able to draw out some of those questions and maybe look at some of the key topics. So we can sort of work on that. So please get thinking. Anything you want to ask, you can email me, john@trees.org.uk. We'll send all this stuff to our panelists and really hope to see you all next week to continue this fantastic discussion. Thank you to our panelists. Thank you to all of you watching out there. And we'll see you next week. Thank you. Bye-bye.
Thank you.